And so what my lab is interested in figuring out is how plant cells use those pieces, the proteins, all the different parts that go into making a biosynthetic pathway, and what information are we missing that we need if we're going to be clever metabolic engineers just like plants are.
We know that many plants make pharmaceuticals, if you will, but what we don't know as much about is how do these plants synthesize these compounds, and how is it that it came to be that plants have evolved the capacity to make this enormous degree of chemical diversity that we don't see in animals, for example.
The cave here allows us to visualize proteins in 3 dimensions using special goggles and tools. Instead of on a flat screen, it's more difficult to see how things align with each other. So by being able to display things in 3D in the cave, it gives an in-depth look in a protein in the cell, along with its substrates and any other interacting molecules.
We've been working for years and years, 25 years, 30 years now, on developing these kind of miniaturized potato plants with a reduced chromosome number.
All of these different plants produce these rather unique natural products. And for human beings, we take advantage of that diversity of natural products that's produced. So, for instance, in my own research, one of the compounds that I work on is the basis for the flavor components of mustard and wasabi and horseradish. And so there's obviously a flavor aspect to that.
What I really want to do is to improve plants in order to make them grow better on nitrogen-poor soil, or make them export more nutrient to the edible part of the plant, so that the people, for example, who are relying on single grain or crop plant as the majority of food source, those people can be in better shape.
And the ones we've developed have only 12 chromosomes, and they're very small plants, very weak, but they've been the key to actually unlocking the potato genome. The potato genome is very, very complex, and there's been a sequencing consortium over the past few years to try and sequence the potato genome. And now you can find just about any gene you want in the potato genome in this double monoploid draft.
By modifying amino acid transport in the plant from the leaves to the seed, we can modify the protein content in the seeds. So soybean is a very good example for that.
So by learning more about how plants synthesize these chemicals, we can use that information to modify the metabolism of these plants to perhaps make chemicals that are more ideally suited for pharmaceutical purposes.
If we can manipulate the levels and composition of these compounds, of these storage compounds in the seeds, then we can alleviate, for example, the energy and food crisis that's occurring in the world.
It's a very interesting evolutionary problem. It's also a very interesting engineering problem, because plants make these things in an incredibly crowded and already very complicated cellular environment. It's very hard to engineer plants or other organisms to make something new, make something different.
Our miniaturized, what's called a monoploid potato, came to the rescue, and it was easy to sequence. So now it's been selected by the consortium to represent the draft genome for potato.
The long-term application of my research would be to modify protein content in seeds, either to increase it if you want more seeds with more protein, or decrease it if you want to increase starch of oil content. Depends on the crop.
And there's also the plant-specific goals, such as improving crops to be more resistant to, drought, blight, things they're already affected to. There's already a lot of considerations with plants, such as how are we going to feed the future over the next 50 years, right? The global population is increasing, and our food supply is staying somewhat finite. So how do we increase plants to meet the demands for future generations?